Okay, I think it's time to start. Welcome to the SmartMat series lecture. My name is Ziyu. I'm from Department of Chemistry in Tianjin University. It is my great pleasure to have Professor Irshad Hussain from the Lahore University of Management Science in Pakistan. Professor Hussain got his PhD from Liverpool University in UK. Now he is a professor in the Department of Chemistry and Chemical Engineering at LAMAS. And he is also the founding chair of Chemistry and Department at LAMAS. Professor Hussain's research is in the nanomaterials. Mainly he tuning the size and the surface chemistry for the nanomaterials to make the application in catalysis, biomedical, and environmental science. Now he has published over 120 peer-reviewed articles in prominent journals like Nature Material, Science Advance, Nature Communications, JACS, Advanced Materials, and Angiochemy, and many other journals. Professor Hussain now is also the chair of a national core group of nanotechnology in Pakistan. He also awarded 02 gold medals in the chemistry by Pakistan Academy of Science. So it's our great pleasure to have Professor Hussain to give his lecture today. So, Professor,
thank you very much,
share your
Professor
screen.
Ziyu. Yeah, I'm going to share the screen. So, can you see that?
Yes, I can see it. Yes, please.
First lecture series of SmartMat. It's really an honor for me. As you mentioned, I work at Department of Chemistry and Chemical Engineering, Syed Babar Ali School of Science and Engineering, Lahore University of Management Sciences, commonly known as LAMS. So here is the outline of my talk today. So briefly, I will be introducing LAMS and my research group, which is Functional Nanomaterial Group at LAMS. Then I will briefly talk about synthesis of metal nanoparticles and nanoclusters, and then their applications in biomedical sciences, renewable energy technologies, and catalysis. LAMS is located here in Lahore, which is the provincial capital of Punjab, which is the largest province of Pakistan in terms of population. And we are just a few kilometers away from our neighboring India. Here you can see over here. And LUMS is relatively new university. It was founded in 1985, and now it is non-profit private national university. Initially, it was started with only one program, that was management sciences, and that is why it is still known as Lahore University of Management Sciences, and LUMS has become the trademark for this university. It's not a big university. We have students lesser than 5,000, and faculty members are slightly over 300. And most of these faculty members, almost entire body, is graduated from USA or Europe, from the top universities. This university is known to be the most prestigious university in the region due to quality of education that we offer over here. And that is why it is a bit expensive university as well. So it is basically expensive for those who can pay the tuition fee. But we have very good financial aid system in which 30 to 40% of our students, whether they are undergrad students or graduate students, they are sponsored by LUMS itself. So only thing that really matters over here, that is merit. So we don't care about financials when we are offering admission to the students. And apart from that, one more thing that we normally take pride of is our national outreach program. And through this program, we try to attract the most brilliant students across Pakistan, especially those students whose family income is very low. And then we invite them during one of the summers at LUMS and introduce them about LUMS programs. And if they are admitted then here at LUMS on merit, we offer them full tuition fee waiver. And apart from full tuition fee waiver, we also offer them a kind of stipend, a decent stipend, so that they can live on campus with that stipend easily. So far, there are over 1,270 NOP scholars who have graduated from LUMS, and many of them, they have studied or they are currently studying in the world best universities like Harvard and MIT. So just imagine a guy from a poor family who can hardly survive. So through this program, NOP program, we trained them, and now they are either studying at the top universities in the world or have already graduated from them. So every year nowadays, we are trying to increase the aid that we offer through NOP, and approximately 100 students now they are admitted almost every year here at LUMS. We have 5 schools at LUMS: School of Business, School of Humanities and Social Sciences, Syed Babar Ali School of Science and Engineering, where I basically work, School of Law, and then we have recently started School of Education. This Syed Babar Ali School of Science and Engineering was launched in 2008 with an idea to bring paradigm shift in science and engineering education in Pakistan. I'm saying paradigm shift because most of the brilliant students after A-level or FSC, that is 12th grade, they would tend to apply for professional degrees like engineering and medical profession. And only leftovers like myself would probably go for the science disciplines. So here the idea was to, which is really very unique here in Pakistan, that students are initially undergrad students. They are admitted in the school, not in the department. And during the first year, they are offered courses ranging from biology, chemistry, computer science, engineering, math, and physics. And at the end of first year, student decides themselves what they want to do, whether they want to do major in biology, chemistry, electrical engineering, or computer science. So recently we have also started this chemical engineering program. So in other programs, we have both BS, MS, and PhD programs. But for chemical engineering, we have just started. We only have BS program at the moment. So due to this kind of setup, we get very few students for science disciplines, roughly around 10 every year for chemistry, for example. But these are the students who are really passionate about chemistry. And due to that, our success rate of our students before they are graduated, before they have completed their BS from LUMS, almost 100% of these students, if they want to apply for higher studies, for PhD, for example, and they get offers from the top universities in the world. So here is just the data for 1 year when about 12 students were graduated and 11 of them, they applied for PhD programs, and all 11 students got offer from the world best universities, including MIT, Chicago, and other Ivy League universities. And this is the case every year since 2012. So almost 8 batches have graduated so far with 100% success rate for fully funded PhD, the admission to fully funded PhD programs in the top universities in the world. So this reflects the quality of education that we offer to our students. So here is just the list of the faculty members in our department, that is chemistry and chemical engineering department. And as I mentioned, we have just started a chemical engineering program very recently. So we have 3 faculty members in chemical engineering at the moment, and we are actively hiring more faculty members in chemical engineering program. So if I broadly classify the research interest of these colleagues, they lie in 2 categories. Most of us, they work on materials chemistry, for example. We try to design new materials and then explore their applications in energy technologies, environment, biomedical sciences, catalysis. And another group work in drug discovery discipline. So Rahman, Shahzeb Salim, Wajur Abbas, and Dr. Saeed, they work in drug discovery kind of domain. So I myself lead this functional nanomaterials group at LUMS, and what we do is to develop new protocols for the synthesis of metal nanoparticles with decent control over their size and shape, and of course surface chemistry, and then use these nanoparticles as building blocks to make variety of these new materials and explore the applications of these nanoparticles and these materials in renewable energy technologies, especially, for example, water splitting, hydrogen storage, biomedical sciences for drug delivery, bacteria detection, and other kind of diagnostic, and environmental remediation. And this is just the summary of some of the selected kind of articles that has been published by Functional Nanomaterials Group in collaboration, of course, with many of our collaborators across the globe. And I'm proud to say that I have very effective collaboration in China, especially with Professor Bian Tan at Huachong University for Science and Technology, that I visit almost every year. Recently, for the last 2 years, I have been visiting as a foreign professor during summer every year. So this is how we prepare these metal nanoparticles. We know that there are a variety of methods that you can use, but the method that we use is very simple, that is based on chemical reduction approach. So in this method, what we do is we start from metal precursors and then reduce them by using certain reducing agent in the presence of certain stabilizer. So this metal ion, for example, is reduced to metal atom, and this will attract other reduced metal atom or metal ions to make this metal nuclei at initial stage, and then these nuclei grow further into metal, bigger nanoparticles. So how we control the size is basically by adding some stabilizer, which can, for example, if they are thiol based or if they have amine groups, they can coat these nuclei at very early stage, and then they stop the growth of these nanoparticles to bigger particles. So in this way, we can definitely tune the size and even shape and finally the surface chemistry of these nanoparticles as well. So once these particles are formed, they still are very small. Their size is smaller than 100 nanometer. So they have huge surface energy, and they again have tendency to agglomerate. So we really need to stabilize them. The two ways we stabilize them is, one is electrostatic stabilization, in which we coat these nanoparticles with some charged molecules like sodium citrate or sodium acrylate, so that all particles, they have similar charge, positive or negative, and due to similar charges, these particles repel each other. And these are molecules are electrostatically bound to nanoparticles and can easily be replaced by a variety of other organic molecules and biomolecules. And therefore, they are really useful for biomedical applications. But if you want to make these particles extremely stable, then you need to covalently attach long chain molecules which have thiol or amine at one end that actually attaches almost covalently to these metal nanoparticles. And due to steric bulk of these coating ligands, these nanoparticles, they do not agglomerate. And actually, you can dry them and keep them for many years whenever you want them. So these type of nanoparticles are really useful when you want to make thin films for a variety of applications, including nanoelectronics and optical applications. So here is just the work that I begin with. Many of you know that citrate stabilized gold nanoparticles are very commonly used for biomedical applications. So here, instead of sodium citrate, we explored that we can use variety of other mild reducing agent and stabilizer like sodium acrylate, sodium oxalate, and malate, and then reduce gold salt into gold nanoparticles. In this way, we were able to prepare particles that range in size from 10 to about 100 or 200 nanometer. And especially this method, sodium acrylate method, is really very good method to control the size of these particles from 10 to 15 nanometer. They are very similar to citrate reduction method, but the particles formed by acrylate reduction, they are much more stable than those formed by citrate reduction method. And we have used these particles as building block to make variety of new materials only because of their better stability over the citrate reduction method. In another case, we use this thioether end functionalized polymer, which have this thioether group and then carboxylate group to make these particles soluble in water. And by controlling the ratio of this polymer to gold in aqueous media, we were able to control the size of these nanoparticles from 1, 2, 3, or 4 nanometer in just one step method. And then because this thioether loosely bound to these gold particles, they can be replaced by alkane thiol molecules, and you can transfer these particles from aqueous media to organic media while keeping their optical properties. Apart from this, in collaboration again with Professor Biantan, they developed these type of polymers which have multiple thiol groups over here, for example, that can complex with gold and other metals. And using these type of polymers, we were able to make copper nanoparticles, silver, gold, and even platinum and other metal nanoparticles which were smaller than 2 nanometer. And we call them nanoclusters because in this size regime, they do not behave as metals, but as semiconductors, and that is why they become fluorescent as well. You can see they can be blue fluorescent, red fluorescent, or even green fluorescent, depending on the number of atoms in these clusters and their surface chemistry. So they are really useful for different biosensing, bioimaging applications, and we have been also exploring their applications in different things, especially for catalysis and multi-drug resistance to kill bacteria which are resistant to a variety of antibiotics. So here is just an example of one of our very recent work in which we produced these silver nanoclusters coated with these aromatic ligands, and they were able to kill bacteria which were resistant to other kind of drugs, and that bacteria is Neisseria gonorrhoeae. So this is kind of multi-drug resistant bacteria transmitted, sexually transmitted infections are caused by these type of bacteria. And these silver nanoclusters coated with these organic ligands, they were really useful to kill these type of resistant bacteria. Apart from this, if you note carefully, these clusters, they have a negative charge, but we know that bacteria surface is also negative charge. So in order to enhance the interaction of these nanoclusters with bacterial surface, we developed silver nanoclusters which were coated with cationic polymers like polyethyleneamine, and they were also able to kill, for example, variety of uropathogenic strains which were otherwise resistant against many antibiotics. And recently, what we have done is, we know that it is even difficult to kill bacteria if they hide themselves in biofilm. So in that case, you need to design materials which can weaken the biofilm, and FAPC amyloidosis is one such pathway by which you can use materials to inhibit FAPC fibrillation, and that can weaken the biofilm, and then you can easily kill the bacteria. So in this case, we examined silver nanoclusters of 2 nanometer and 10 nanometer particles, and we noticed that these 10 nanometer particles, surprisingly, they were more efficient to inhibit FAPC fibrillation and to weaken the biofilm. So their antibacterial activity and FAPC fibrillation inhibition were basically correlating with each other, not for these 2 nanometer silver nanoclusters. And then we also have reviewed recently the materials, the chemistry of basically materials that can be used to kill resistant bacteria. And in this case, we highlighted that apart from the nature of materials, what we need to focus is the surface chemistry of those nanoclusters or nanoparticles that we use to kill resistant bacteria, because we know that gram-positive and gram-negative bacteria, they have slight difference in their surface chemistry. So you need to design these materials in a way that they can efficiently interact with either gram-positive or gram-negative bacteria, and they can overcome the pathways that bacteria adopt to develop the resistance against different drugs. And for that purpose, the materials which produce reactive oxygen species and inorganic nanomaterials are among those type of materials which produce this reactive oxygen species, and bacteria so far they are unable to develop resistance against those type of materials. So they can be really useful to kill resistant bacteria, whether they are gram-positive or gram-negative, if you really control their surface chemistry and the nature of material that you use for that purpose. So here is just one slide from my collaborator, Professor Rutello, at University of Massachusetts. They actually designed cationic gold nanoparticles as nanoprobes to detect different type of diseases. They call it chemical nose, basically, that can be used to detect variety of different diseases at very early stage, especially cancer. And they also used these cationic gold nanoparticles to detect bacteria. So how here in this case, for example, you see this beta-gal protein, which is a large protein. When it is coated on these cationic gold nanoparticles, it loses its biological activity and is unable to convert this yellow substrate into red product. But in the presence of bacteria whose surface is negatively charged, these nanoparticles preferably go to those bacteria surface, and this beta-gal protein is released that can then convert this yellow substrate into red product. So in this way, you can develop colorimetric method based on these protein and cationic gold nanoparticles to detect bacteria in drinking water. But note that both these proteins and gold particles, they are expensive. So we wanted to replace these gold nanoparticles with iron oxide nanoparticles. And you will be amazed to note that redox properties of these iron oxide nanoparticles basically mimic the redox property of many enzymes, especially horseradish peroxidase enzyme. And they can convert, due to their redox properties, variety of these yellow substrate, for example, in this case, pyrogallol, into red product, only by adding these iron oxide nanoparticles. But if you control the chemistry of these iron oxide nanoparticles by coating with dopamine, for example, in this case, so that they become positively charged and interact strongly with bacteria. So in the presence of bacteria, for example, iron oxide nanoparticles will be coated by these bacteria, and then it will not be able to convert this yellow substrate into red product. So we designed these nanoparticles, iron oxide nanoparticles, highly stable, and then control their surface chemistry by coating these dopamine ligands to figure out how useful they can be to detect bacteria in drinking water. So in this way, we were able to detect bacteria in drinking water, but unfortunately, their sensitivity is not that great that they can be commercially employed for this purpose. In another case, we also coated these nanoparticles with anionic ligands like this one to render them anionic, to have negatively charged kind of surface. So if they have negatively charged surface, then you can coat them with large kind of, for example, cationic polymers, so that their catalytic activity is reduced, inhibited, and in the presence of bacteria, these bacteria are negatively charged. They will replace these cationic polymers from nanoparticle surface and release these iron oxide nanoparticles. But even again, in this turn-on sensing kind of strategy, it was not really very useful to enhance the sensitivity of this method. So we are still trying to figure out, to basically design even new ligands that can improve the interaction of these nanoparticles with bacteria and therefore enhance the sensing ability, detection ability of these nanoparticles for bacteria. So this cartoon, I believe many of you are aware of this. So here is just to show, it is just a cartoon to show the potential of gold nanoparticles, which are 25 nanometer particles, to detect the cancer at very early stage and then simultaneously treat it as well. So these are 25 nanometer particles, for example. Once they are injected, they travel in the blood vessels, and pore size of blood vessel is 5 to 10 nanometers, so they do not normally come out of this. But where there is a developing tumor, we know that cells multiply very quickly over there. And therefore, these blood vessels become porous over there, angiogenesis. So these particles go out from these pores and accumulate in the region where there is a developing tumor. And then you can use NIR camera to basically locate where these nanoparticles are accumulated. And then you can, of course, increase their local temperature to kill the cancer cells as well. But the problem, it is not that so simple. So you need to perfectly control the surface chemistry of these nanoparticles and then coat them also with the ligands that can direct them to the cancer cells. And for this purpose, for example, to avoid the immune system, what people do is usually coat them with hydrophilic ligands like polyethylene glycol, so that there is a water droplet around this as they are injected in the blood, and immune system is unable to recognize that if foreign particle has been injected. So surface chemistry of these particles is really important if you want to use them for drug delivery applications. And more recently, there has been a lot of work on the size effect of these nanomaterials, and it has been found that particles which are between 50 to 100 nanometer, they are really useful for these type of applications. But if you use 50 to 100 nanometer particle, they have smaller surface area compared to if you have collection of these nanoparticles or assembly of these nanoparticles of the same size. Or in other words, if you can make this particle porous in nature, so then you can deliver more drug by using minimum amount of this inorganic material, because there are issues related to the toxicity of these materials as well. So you need to deliver as small amount of these particles as possible. And for this purpose, we have designed variety of new protocols for the synthesis of porous gold nanoparticles, lipid nanoparticles, and even hybrid, organic, inorganic hybrid nanoparticles for these type of applications. So I'm not going into detail of these kind of applications because of limitation of time. And here is just our recent perspective on the coronavirus. We know that is currently the serious pandemic globally. And what we have highlighted in this perspective is the use of lipid nanoparticles to deliver RNA, for example, that can inactivate the virus in the lungs. And through this, we have proposed this inhalable treatment for human respiratory viruses. So these lipid nanoparticles, which are hollow basically, so you can encapsulate RNA over there, and that RNA can bind with the viruses in the lungs, for example, if they are inhaled and that they are inactivated. So there is a lot of actually interest in this type of work very recently. So this was all about biomedical applications of these materials. Apart from this, we are also exploring the applications of these materials, especially nanoclusters, for renewable energy technologies. And among them is mainly a kind of water splitting to produce hydrogen gas, and also little bit about solar cell technology. So in solar cell, for example, dye sensitized solar cells, we need to use some dye sensitizers that can absorb major portion of sunlight. And we know that these ruthenium-based dyes are very common for this purpose, but this ruthenium organometallic complex, we know it is very expensive, and it is also there are stability issues as well. So in 2009, Professor Stilachi group, he is also my collaborator. Initially, he was working at MIT, they proposed that if they can control the size of silver nano clusters to only 25 atoms, so these silver nanoclusters will not behave as metals, but they will start behaving as semiconductors. Their HOMO and LUMO will be separated, and due to this HOMO-LUMO transition, they can absorb major portion of sunlight, like dyes molecules. And in fact, they were able to prepare these silver nanoclusters using these kind of ligand, which is simply mercaptobenzoic acid. And you can see that they were right, that it absorbed major portion of sunlight. But unfortunately, they were not very stable. So they lose their optical properties within 24 hours. So luckily, I was visiting his group during the same period, sometime in 2010. I came across this problem, and when I came back to LUMS, we started exploring how can we improve the stability of these silver nanocrystals. And for that purpose, we used these kind of ligands, which in addition to this carboxylic group also have nitro group. Basically, it was our finding that if you increase the hydrophilicity of these nanoclusters, they will be more soluble in water, in aqueous media, and they will be more stable as well. So in this way, we were able to make highly stable silver nanoclusters. They were extremely red in color. They absorb in UV and visible region, but unfortunately, the intensity of absorption was not that great. So we are now working on designing new ligands to make not only these silver nanoclusters which can absorb major portion of sunlight, but also their alloys with other metals, especially gold, and see how this allying affect the optical properties of these type of materials. Apart from this, very recently we got more interested in transition metal oxide nanoclusters. Nanoclusters, I mean, these are nanoparticles which are smaller than 2 nanometer, and then explore their applications in, for example, water splitting for hydrogen production. We know that water can be splitted to produce hydrogen gas, but energy input in this case is unfortunately more than energy you will get from this produced hydrogen. And the thermodynamic and kinetic barriers basically require a potential much higher than this 1.23 volt potential, that is the minimum potential required to split this water molecule into hydrogen gas. So there is a need to develop catalyst which can reduce the thermodynamic and kinetic barriers to split water into hydrogen gas, so that it can be splitted to produce hydrogen at an economical cost, and so that it can be practically used as well. And for this purpose, we have worked on a variety of new transition metal and metal oxide nanoclusters. For example, we started with nickel nanoclusters. In this case, you simply reduce nickel with the carbon monoxide in the presence of phenyl ethanethiol, and by controlling the ratio of this phenyl ethanethiol and nickel, we were able to produce atomically monodispersed nickel nanoclusters that contain either 4 nickel atoms or 6 nickel atoms. And here you see this optical absorption is not due to the surface plasma resonance band as normally you would see for gold or silver particles, but it is due to their optical transitions of HOMO-LUMO, because they behave like almost metal molecules over here. They're so small that it is really difficult to characterize them using even high resolution transmission spectroscopy. So we need to use MALDI, for example, mass spectrometric techniques, and single crystal XRD. If you are able to crystallize these kind of nanoclusters, you can then characterize them by single crystal XRD. So in this way, we were able to find that they have either 4 nickel atoms or 6 nickel atoms. And interestingly, they were able to reduce those thermodynamic and kinetic barriers for the splitting of water. For example, they were able to oxidize water at 1.51 volt versus reference hydrogen electrode potential. But problem with these nanoclusters, because they were really soluble like metal molecules, you cannot reuse them. So to make this process really more economical and practically feasible, you need to have these clusters on various spores. And for this purpose, what we did recently is we functionalized graphene oxide with thiol. So we made use of epoxide groups on graphene oxide, and then converted that into thiol group by a series of different chemical reactions. And then we generated these nickel nanoclusters on this. And these silver nanoclusters again were smaller than 2 nanometer, and they were really quite active to oxidize water to produce hydrogen. Here you see onset potential in this case is 1.46 volt, which is really very decent, and they have quite low kind of Tafel slope as well. And more importantly, they were really stable at least for 3 days. They for continuous CV, they maintained their stability and activity at least for 3 days. In another case, what we did, we used these nickel nanoclusters and nanoparticles as catalyst to grow nitrogen-enriched carbon nanotubes. And depending on the size of these nickel nanoclusters or nanoparticles, we were able to control the thickness, the diameter of these nitrogen-enriched carbon nanotubes as well. And we found that these are really useful to, again, split water. And they were useful not only for OER, oxygen evolution reaction, but also for hydrogen evolution reaction. So they are kind of dual catalyst and very useful for this type of application. Apart from this, very recently, we also coated these kind of graphene oxide nanoparticles with the copper and cobalt oxide nanoclusters and self-assemble them in the form of these small chain-like structures on the graphene. And we found that they are really very, very stable, highly stable kind of catalyst, which can work under extremely alkaline conditions to produce hydrogen gas from water by the splitting of water. And here in this case, we generated these cobalt nanoclusters in nitrogen-enriched mesoporous carbon network. So this is mesoporous carbon, and within these pores, we generated cobalt nanoclusters. And again, we found that they were really extremely stable over here. So up till even 4 or 5 days, they maintain their catalytic activity for OER reaction, oxygen evolution reaction under alkaline conditions. So all these materials indicate that these transition metal oxide nanoclusters, they have really great potential to minimize the energy barriers that are required to separate water to produce hydrogen gas. So once this hydrogen gas is produced, you can either use them immediately or you need to store that. And in order to store that, we also worked in collaboration with Professor Biantan and here at Hwa Chong University for Science and Technology. And we exploited, in this case, a Friedel-Crafts alkylation reaction. So chemists would know about Friedel-Crafts alkylation reaction, in which aromatic compounds can be alkylated in the presence of Lewis catalyst. And one of the limitation of this process is polyalkylation. And we, in fact, made use of that limitation to basically polymerize variety of aromatic monomers into microporous polymers. And these microporous polymers, they have huge surface area, even more than 3,000 meters square per gram, and they have ability to store carbon dioxide and hydrogen. Decent actually storage capacity for both of these two gases. And moreover, even more recently, what we did, again, mainly by Professor Bian Tang group, that they coated titanium nanoparticles with these microporous hypercross-linked polymers and then immobilize them on graphene oxide. So, for example, carbon monoxide, that can diffuse through these microporous polymers, which are coated on titanium nanoparticles, and guide them to titanium nanoparticle surface, where they are photochemically converted into hydrocarbon fuels. So in this way, they were able to convert carbon dioxide into methane gas. So this is very actually a kind of trending research these days, how you can make good use of carbon dioxide to reduce greenhouse effect and simultaneously make good use of that by converting that into hydrocarbon fuel. Apart from this, we also generated these kind of metal nanoclusters of palladium and platinum in microporous hollow capsules. And by confining these metal precursors in the smaller pores of these polymers, we were able to produce very small kind of clusters in these polymeric capsules that were catalytically active for Suzuki coupling reaction. We know that Suzuki coupling reaction is commonly used for carbon-carbon coupling reactions when you want to develop new carbon-carbon bonds. But for that purpose, normally use precious metal organometallic complexes as catalyst. And we know that they are very expensive and they cannot be reused as well. So these type of catalysts, they were very effective, and meanwhile they can be reused as well. So they could be very useful for this type of chemistry. Apart from this, we also worked in collaboration with KAUST group. KCC1 is KAUST Catalysis Center 1. This was the first actually material that this center developed for their industrial applications. This is nanofibrous kind of silica that has huge surface area. What we did is we functionalized this silica nanospheres with amine groups and then generated very small clusters of gold, which are 1 to 2 nanometer, and then here in this case gold particles 4 to 6 nanometers, and then 10 to 15 gold nanoparticles. And we try to figure out how the size of these gold particles affect the catalytic properties of these kind of heterogeneous catalysts. And we figured out that smaller is the size of these gold nanoclusters. In this case, more effective they are to catalyze the conversion of carbon monoxide into carbon dioxide. This is again another kind of strategy in which we used gold nanoparticles as building blocks to make new materials. For example, in this case, these are polyacrylamide beads, which are hierarchically porous. You can see these macropores, and within these bigger pores, there are smaller pores. So it is basically hierarchically porous polymer bead. So when we soak this in gold nanoparticles, so these were the acrylate stabilized gold particles. The citrate were not that effective. because they would simply agglomerate at the surface. So these acrylate gold particles were stable enough that they can go into these pores, they can infiltrate and within these beads, and when they are completely loaded with these gold particles, you can calcinate to remove the organic polymer and have these pure gold beads. So this is 99% gold, pure gold, and you will see that these gold beads retain the emulsion templated porous structure of these polymer beads. So in this way, we were able to reproduce the emulsion templated porous structure of polymer bead into emulsion templated porous structure of these gold beads that have decent surface area. And due to this higher surface area, they can be very useful catalyst, and as well as they can be useful for biosensing applications. You can develop kind of electrodes using this porous gold material. And we then extended this to other metals which were catalytically more active, like palladium and platinum as well. So basically, this is general strategy that can be used to make these kind of porous metals using metal nanoparticles as building blocks. We further extended this work in this case to develop a generic strategy to produce heterogeneous catalyst again. So in this case, we have polymer gold nanoparticle composites. It is still highly porous, and you can infiltrate variety of inorganic soul into this. In this case, it is silica soul, but you can equally use silica, titania, zirconia, or aluminium soul. And once you have this composite, then you can calcinate to have these gold particles on silica, in this case, silica matrix. So in this case, we used 15 nanometer gold particles, but because this is very generic strategy, you can change the matrix from silica to any titania, zirconia, or alumina, and you can also control the size of these nanoparticles, not only for gold, but it also works for other metals as well. And then more recently, we have extended this approach to develop a variety of new emulsion templated polymers in which you can control the chemistry of polymer matrix, and then you can incorporate a variety of inorganic nanoparticles as well, especially magnetic nanoparticles, silver nanoparticles, and other metal nanoparticles. And because of their higher surface area and porous nature, we use them to remove organic and inorganic pollutants from wastewater, for example, which are contaminated with organic kind of contaminants or inorganic metals, for example, toxic metals. So this was really useful, and these all applications, the publications are basically from these type of materials. And lastly, we also developed this very simple directional freezing approach by which you can use the concentrated, basically, dispersion of nanoparticles, could be metal oxide, or even polymer nanoparticles. And you can freeze them directionally, and by freeze evaporation, freeze dyeing, then you can make variety of these new materials depending on what was the material in which they were freezed. And this, for example, fishbone-like structure, this is from polyvinyl alcohol, and we found that this is very useful for tissue engineering applications because cells can grow very happily over here. So this was again in collaboration with Professor Yang, Haifa Yang from University of Liverpool when I was doing PhD over there. And apart from this, we also developed this very simple technique by which we can cross-link these gold nanoparticles as monomers using these bifunctional ligands, dithiols. And by controlling the ratio of these dithiol to gold nanoparticles, By the way, again, the particles we used over here, they were acrylate stabilized because we need to disperse them in ethanol. So citrate stabilized gold particles are not soluble in ethanol. They would immediately agglomerate. But these acrylate stabilized gold particles, they were stable up to 90% of alcohol. So we dispersed them in 90% alcohol and then added this. And by controlling the amount of this linker, we were able to make either dimers or trimers or even oligomers of these nanoparticles. And we can grow these nanoparticles stepwise. And you will note that as we grow them, their optical properties changes like this. So initially they have a plasmon resonance band at 524. So as they form dimers, trimers, they develop this longitudinal band over here. And this is typical, actually, optical kind of spectrum for gold nanorods or gold nanowires. And by cryo-TM, we confirmed that these actually nanowires, chain-like structures, which are up to micrometer long structures of gold nanoparticles, they exist in the solution, basically. And in this way, you basically tune the optical properties of these nanomaterials, and we believe that they're really useful for optical waveguides as well. So this was all summary of our work so far. And again, I would like to mention that this was done in collaboration with many of my collaborators across the globe, in USA, in Europe, especially in UK, Germany, in Switzerland, and more importantly, in China, especially with Huazhong University for Science and Technology and Kengabu University for Science and Technology. So I would like to thank all these groups and these kind of institutions and funding agencies who have supported all our purpose. And thank you very much once again for listening.
Okay.
So I'm available if you have any questions. So I believe I was on time, right?
Yeah, of course.
Yeah.
It's a very great talk. I think probably I should start asking questions. Actually, I have many. Okay, so first thing, very interesting about the Nico nanoclusters. You can have two sites, mono distribution, two different sites. One is Nico cluster with 4 atoms. The other one has 6 atoms, right?
Yes.
How can you keep such a good mono dispersion? Because usually nanoparticle you get the multi distribution for the number of atoms in the particle. So how is this possible?
Yes, very interesting question. Yes, I agree that it is really difficult to control the size of nanoparticles. So to make them atomically monodisperse is a real challenge. But when we talk about nanoclusters, which are smaller than 2 nanometer, and where you can, so that can only be done if you have single crystal of these materials.
Wow.
And you cannot really crystallize if they are polydispersed. So they must be atomically monodispersed, and only then you can crystallize these kind of materials.
That is really amazing. Yeah, it's very hard for me to imagine that it's possible. Okay, very nice.
Yeah, there is actually a lot of work being done in nanoclusters for this type of purpose.
Okay. Yeah. Okay, I got some message from the other from the audience who watch the live broadcasting. one person asked that can you tell a bit more about the using nanoparticle to deliver the antivirus for the antivirus purpose of you mentioned about the nanoparticle for the SARS and the COVID virus. Can you tell a bit more about how the nanoparticle can work as the antivirus medicine?
Yeah, in that case, what we highlighted is basically to use liposomes. And we know that liposomes are hollow kind of lipid bilayer, which have hollow interior. And in that interior, you can encapsulate the viral RNA that can bind to the genome of COVID-19, for example, specifically. And if you then deliver this through inhaling, that RNA will bind to viral genome and inactivate that. So that is just kind of a suggestion, a kind of idea that need to be explored, because we know that gene therapy is also very commonly used to inactivate the expression of various genes. So it is very, very similar to that, basically.
Okay. Probably I don't know.
But we know that RNA is highly unstable.
Okay.
So we really need to develop the strategies by which you can deliver this RNA in a stable form, in active form.
Mm-hmm. Okay. Let's see. Another question asked about also about the nano cluster. The question is how the nano cluster work as a catalysis when there is a ligand on the cluster.
Yeah, actually, ligand. There is a lot of work being done to figure out how this ligand affect the catalytic properties. So definitely, if there is ligand on the surface, it is completely coated, its catalytic property will be hampered. But for catalytic applications, you need to, once they are immobilized on the desired substrate, you need to remove that ligand. And there are various ways you can remove that ligand by, for example, ozone treatment or so many other treatments that can be used to remove those organic ligands from nanoparticles. So yes, if these are bare kind of nanoclusters, nanoparticles, they are more effective as a catalyst. So most of the work that we have done, for example, in case of immobilized nickel nanoclusters or copper or cobalt, for example, so those are the catalysts that were treated at high temperature as well. And in that case, we did not use any ligand to coat their surface. We simply generated those nanoclusters on functionalized graphene. So graphene is oxide acting as a support, and then bare kind of nanoclusters sitting over there through this thiol metal bonds.
Okay.
So yes, these ligands hamper the catalytic properties, and they need to be removed for catalytic applications.
Okay. One of my friend asked he's interested in the national outreach program you ever mentioned at the beginning of your lecture.
Yes.
Is the name national outreach?
Yes, it is the National Outreach Program, NOP.
Yeah. He's asking, is there any possibility like the university in China can be participate into this program? I think it's a very interesting question. Yeah.
Yeah, it is really interesting question. So basically, this NOP program is for people with low income. Yeah. For example, what we do is those families whose monthly income is less than 50,000 rupees.
Which is how much Chinese?
They can 50,000 rupees is about $300 per month. So if family is having this much income, they are eligible to apply for this purpose. So if any university from China is interested, then I can certainly connect them to our administrative offices over here at LAMS, and they can see how can we do it together.
Yeah, probably he said it's an interesting program that Chinese university can use for the recruitment of excellent international citizens from Pakistan. I think that's an interesting idea.
Yeah, if you have any such program which is fully funded, and you also offer stipend for undergrad education, so that can, I think, be discussed, because we normally every year invite hundreds of these students at LAMS, and we actually give them some idea about LAMS, and then offer them crash courses about different subjects being taught here at LAMS. So if there is any interest, probably your university can collaborate with LAMS and jointly identify the students who may be interested to go to China for undergrad studies.
Is there any scholarship?
But that is for only poor families. They won't be able to bear the expenses.
Okay. So do you have any scholarship that provided by other universities outside Pakistan to promote the exchange or the collaboration between the undergraduate students in LAMS and the other universities?
I think yes, our university has signed MOU with a number of universities in different countries, and student exchange at undergrad level and graduate level is possible. Yes. So, but for that purpose, we need to discuss this with our international office. And one of my friend, Professor Wakar, is basically heading that international office. And if there is any interest, I can talk to him.
Okay. I think we already have many questions answered by Professor Hussain, and I think it's time for us to call for the end of this lecture. And let's thank Professor Hussain again for those wonderful talk. Okay. Thank you.
And thank you, Ziyu.
Yeah. And we would like to thank all the audience to join this.
Thank you to all the audience, wherever they are.
Okay. Thank you.
Okay.
That's the end of this presentation. Thank you. Bye-bye.
Bye-bye.